it's interesting you were concerned with the video, because that's basically what we're sort of arguing about. The AV person wanted us to do it via Zoom, and I preferred it via not a Zoom thing, but a whole wire so that it's works smoothly, you know?
Yeah.
Okay.
Oh, this is a photo shoot, or?
Yeah, okay. Yes. We are very much, communication PR. Yeah.
All right. Yeah, sounds good.
Yeah.
Okay. All right. Perfect. So we move our mask or?
Yeah.
Okay.
Very still. Okay, one more. I'm sorry. Okay, great.
Yeah.
Thank you. Thank you.
Thanks. Thanks, Dan.
That's an awesome.
Yes. Okay, I think we are ready to start. Welcome, everyone. Welcome to our BME seminar series, which is hosted by the Department of Biomedical Engineering at Columbia University. My name is Organoids-on-a-chip. I'm the Diversity and Microbial Foundation Professor of Biomedical Engineering and Medical Sciences here at Columbia. Today, we continue to offer this lecture in a hybrid format with both virtual and live audience. Our speaker will present for approximately 40-45 minutes. Right?
Okay.
And we will have about 15 minutes at the end for Q&A. For our virtual audience, throughout the presentation, you can use the Q&A feature, not the chat, the Q&A feature at the bottom of your screen to submit your questions. This is obviously for people who are remote. Here, you just raise your hand. We will try to answer as many questions as possible during the Q&A session at the end. I think this is all we needed. Okay, so today we are really excited to welcome Professor Dan Ha. Dan is Associate Professor at the Department of Bioengineering at the UPenn. And he's a real pioneer of organ-on-a-chip technology. His research group at Penn is focusing on developing microengineered models of human physiological systems for biomedical and environmental applications. His particular claim of fame is this landmark paper, Lung on a Chip, which really has defined and continues to define the entire field. He has won, not surprisingly, very many honors and awards, including the Bernard Langer Distinguished Lectureship, Lush Prize, the McPherson Distinguished Lectureship, CRI Technology Impact Award, John Ryan Medal, Design of the Year Award, and Best Product of the Year Award from not less than London Design Museum. So his designs are also very pretty. And he received the NIH Director's New Innovative Award, which we all know is extremely prestigious. So Dan, welcome, and we all look forward to your lecture.
Well, thank you very much for the introduction, and I would like to start by thanking Gordana and the department for the invitation to speak about our work today in person. I think that's what makes this visit special for me. And hello bananas, they're up there. So all right, so we have a lot to cover, and actually we had some technical difficulties at the beginning, so I'll try I'll just get started and try not to waste any time here. Just as a quick introduction, this is my lab at Penn. We're basically a group of bioengineers working on something called organs-on-a-chip. So I'm sure many of you have heard of this, and also Gordana is a true pioneer in this area as well. But just to kind of for those who are new to this, so basically research in my group revolves around this idea of using these microfabricated devices for cell culture. But we also use micro and nano engineering technologies to mimic complex structure and also very complex and dynamic environment of human body in ways that have not been possible using traditional cell culture model systems. And so if you think about it, the idea is to use these systems as micro-engineered models of human tissues and most functional elements of human organs for a wide variety of applications. So what I'd like to do today is to give you a quick introduction to this technology by talking about some of the design principles that we use and try to actually help you better understand what this technology really allows us to do or may allow us to do in biomedical research using several examples of organ chip systems that we are developing in my lab at Penn. So that's what we're going to do today. Okay, so if you're new to this, one of the first questions you could ask is, how do we actually make these organ chip systems? How do we go about creating and designing these organ chip model systems? So I'd like to just spend just a couple of minutes talking about the design principles that we use in constructing these model systems. So obviously you would have to have a target organ, and for this discussion, let's say we're interested in mimicking the lung and developing lung on a chip. So in this design process, the very first step is to break down your target organ into the most critical functional elements that we call functional units. So in the lung, those would be the alveolar system shown here. This is where a lot of important things and interesting things happen. So once you identify the functional units, then you closely examine those units to identify the key features. So we often start this process by looking at the structure and architecture. So what I'm showing here is a section between alveolus and a surrounding blood capillary. So there's a very thin layer of alveolar. Maybe it's better for me to use this actually for those who are joining us on Zoom. So there's a very thin layer of alveolar epithelial tissue on one side and another thin layer of capillary endothelial cells separated by a very thin interstitium. And so during this design process, we also pay a very close attention to environmental cues, biochemical, biomechanical cues that those cells and tissues see in those units. So in the case of the alveolar system, what's really interesting is that, as you can see in this animation, alveolar sacs like balloons, they expand and contract during respiration. There's airflow on the lung side, blood flow on the vascular side. So once you identify those key features, the next step is to go ahead and design the system that would allow you to mimic and replicate those features as much as you can. So this is actually a proof of concept kind of study that Gordana briefly mentioned that I worked on as a trainee with Doningberg at Harvard many years ago. But I think it's a good example I could use to talk about design principles and proof of concept of this technology. So what you're seeing here is a device called breathing lung on a chip. So the animation shows the cutaway view of this device. So there are two chambers at the center separated by a thin porous flexible membrane, and we can culture lung cells on one side, capillary cells on the other side to mimic that architecture we just saw, right? And using this engineering design where we have those hollow cavities on, right next to the cell culture chambers, we can apply cyclic vacuum suction to stretch the tissue layers to mimic breathing motions, in the system. So I hope you get the idea, right? So using these devices, we can, culture many different cell types at the same time to mimic structure and the environment. But I would say it's far more than that. So by having these, multiple cells talk to each other in the same device in this physiological environment, we can start thinking about mimicking more complex, responses and functions that happen at higher levels of organization, not just a cellular level, but the tissue and organ levels. So to help you better understand what I'm talking about here, let me actually use this, infection as an example. So let's say your lungs are infected with bacterial cells. And as many of you know, one of the really interesting things, important things that they do is they stimulate alveolar epithelial cells to produce and secrete a lot of pro-inflammatory cytokines that actually, diffuse into the surrounding tissues. And when these cytokines reach the blood vessels, they actually activate endothelial cells lining the capillary vessels. So I use this movie to help engineers to better understand how this process happens. So let's say these neutrophils, white blood cells circulating in your body, patrolling the body. So they have the ability to recognize those, activated endothelial cells. And so what they do, as shown in this animation, they, engage the endothelium, and then they actually stick and roll, and eventually they squeeze themselves through the capillary tissue into the alveolar air sacs. And once inside the air sacs, they start crawling directly towards the site of infection. This is not exactly not how they crawl, but when they get there, they actually try to resolve the infection by engulfing those bacterial cells. So this is actually how our lung fights off infection. And as far as I know, this is one of the most complex organ level, not tissue or cell level, organ level responses, integrated responses that happens in the respiratory system. So the question is, can we mimic this in this man-made system? So the first, experiment we tried was to flow, white blood cells, fluorescent intact white blood cells in just a normal, model. As you can see here, nothing interesting happens. There's a zip by. But when in the presence of bacterial cells on the lung surface, they now stick, presumably because those endothelial cells are now activated. And this is my favorite movie. That it shows the entire process. It sticks, curls around. At some point, it starts, wiggling its way through the capillary cells in one of the pentagonal pores on the membrane to get across the tissue layers from the lower vascular compartment to the upper lung chamber. And once inside the lung chamber, these white cells shown in red, they chase the bacteria and phagocytose them, just like what happens in the living human lung. So here, what I'm showing here, if you think about it, there's nothing scientifically new here, but being able to mimic this multi-step cascade, very complex process, and being able to watch, this process happen in real time at high resolution, I think that's a great actually thing to have here. Yeah. So, this, like I said, was a proof of concept demonstration that we did, many years ago. But if you think about it, you can use exactly the same microengineering biomimetic approach to mimic any other organs, in the human body on a chip. So let me actually show you an example, a more recent example developed by my lab. So we have a model of the eye. In this case, we're mimicking the front of the eye called the ocular surface. So for those who are new to this, ocular surface basically refers to one of the functional and structural units of the eye, consisting of the cornea at the center surrounded by the conjunctiva. So these are what the tissues look like. So corneal cells tissue contains this stratified epithelium supported by this collagen-rich stroma and also it contains the cells called keratocytes. And conjunctiva epithelium contains these goblet cells that produce mucus, which is one of the key components of tears. And this is something we as engineers found really interesting. So, blinking, as you can see in this animation, is very important for spreading tears, on the surface to keep the surface hydrated, and also it forms a nice refractive surface for light transmission. So we wanted to mimic those essential features, kind of following the same design principle. This is actually what we came up with. It's called the human blinking eye on a chip, developed by this excellent former grad student of mine, Jung Yun-seo, in collaboration with these three ophthalmologists shown here. So this is what the entire device looks like. We use 3D printing based, technique to actually produce these, dome-shaped scaffolds that look like contacts. It's a very highly porous, scaffold. So to mimic these tissue layers, we, for example, mix collagen with keratocytes. We inject the mixture into the cavities of the scaffold. As you can see here, this image shows a nice deep penetration of these cells in the scaffold. We also came up with the way to deposit these two different cell types: corneal cells at the center, surrounded by the conjunctival cells, using special surface patterning technique. So it's all about giving the cells the right environment. In this case, physiological environment. So what we do is we first form a confluent monolayer of cells on the surface of the scaffold, and then we expose these cells to air to induce differentiation. Because if you think about it, that's the environment these cells see, tissues see in the body, right? So as a result of this ALI, air-liquid interface culture, corneal cells that start off as a single layer become stratified, forming multiple layers. They express tight junctions. They enhance their terminal differentiation markers like cytokeratin 3 and 12. And initially, their surfaces are flat, but as a result of differentiation, they form these beautiful structures, microvilli, microplicae, that are almost indistinguishable from those features seen on the surface of the human eye. And we've also confirmed that some of those conjunctival cells differentiate into mucus-producing cells in this device as a result of differentiation. So then, okay, we can engineer the tissue. Then what about blinking? So this is actually the setup where we use this biomimetic eyelid made out of gelatin. And it's actuated by this electric motor. So this actually hydrogel layer slides back and forth over the surface of scaffold to mimic blinking. So here's a zoom, kind of a close-up of what happens. So this is a tear secretion channel into which we inject contrived tears, artificial tears. We apply a little bit of pressure, fluid comes out, it gets picked up by the sliding eyelid and it gets spread on the surface. So as a result, we can form tear film. Actually, we used OCT, optical coherence tomography, to actually visualize and measure the thickness of the film. It was about 6 microns. And according to the literature, physiological tear film thickness ranges from 5 all the way up to 10. So this falls within the physiological range. In collaboration with Vivek Shenoy at Penn, we also developed a computational model of the system to kind of understand blink-induced mechanical forces. And so we modeled the eyelid and then the tissue as deformable structures, and then we also consider this tear flow in this thin gap between the eyelid and the scaffold. So using this computational model, we were able to estimate and also map blink-induced mechanical forces. For example, fluid shear stress exerted by blinking eyelid, and also deformation of the tissue. So interestingly enough, there are clinical papers published 10 or more years ago where they actually used miniaturized sensors to measure those things. And as you can see in this table, by comparison, our computational predictions were in excellent agreement with those clinical measurements. So at least we can claim that this system actually allows us to form physiologically relevant biomechanical environment in this man-made system. So talking about mechanics, motions, initially we were very concerned about cells sloughing off due to blinking, but as you can see here, the live cells are green, dead cells are red. You hardly see any dead cells even after repeated cycles of blinking. Cells remain adherent and viable. But it was more interesting thing was so this is actually one of the most interesting findings from this study. So we found out that blinking actually allows these cells to differentiate more quickly. So if you look at these, the expression of these terminal differentiation markers in those cells cultured at the ALI alone, if you compare these guys to these cells, that were subjected to blinking, we saw, significant increase in the expression of these markers. And so based on these results, we're speculating that the physiological mechanical forces, might be contributing to cell differentiation, cell maintenance. So, we're thinking that this blinking might have a biological role. Okay, so I hope those two examples, lung-on-a-chip, eye-on-a-chip, give you a better sense of how organ-chip systems can be engineered using human cells in these microfabricated devices. So I guess now that we know how sausages are made, so the interesting question you could ask is, what are the current applications of organ chips, right? So over the last 10 years or so, people actually have been working to develop disease models using this technology and use these model systems for mechanistic studies or for drug testing to develop better, safer, more efficacious drugs. So let me actually give you an example of these types of studies using this blinking eye-on-a-chip system that I just talked about. So in this work, actually, we became interested in modeling dry eye disease. So it sounded very simple. We thought we would have to just keep it dry and we'll be done. But as it turns out, it's an amazingly complex disease. As you know, it's a very prevalent disease, but there are many different subtypes. As of today, there are only 2 FDA-approved drugs for treatment of dry eye in the United States, and there are a lot of outstanding questions. So can we use this technology to kind of improve, clinical treatments or our understanding of the disease? So one of the first things we had to do was to design this chamber where we could, maintain this device at the relative humidity of room air, not 100% humidity of, conventional incubators. So this is a computer-controlled system. We have a feedback, sensing system here to maintain the humidity. To mimic the dry eye, we actually reduced blinking frequency by half. So it may seem kind of extreme, but as it turns out, this is one of the most common causes of dry eye disease, actually, especially the evaporative type of dry eye disease, known in the literature. So I said, there are many subtypes, in dry eye, but regardless of the type, there are two core mechanisms. Okay, this is our current understanding. So the first mechanism is called tear hyperosmolarity. So as you can imagine, as water evaporates quickly from the tear film, salt concentration in the tear film increases dramatically, and that's known to induce ocular inflammation. The second mechanism is tear film instability. So water evaporates and tear film actually becomes unstable, and this leads to premature breakup of tear film in those patients. So we wanted to see if our model was capable of emulating those core mechanisms in vitro. So to measure osmolarity, we use this handheld clinical instrument called Tear Lab to sample tears from our devices. It's actually nice thing about this model is it's big, so you can actually use those clinical measures, for direct, analysis. And so we saw, a lot of similarities between the eye-on-a-chip and the human model, both in normal model and also DED. This is actually a really cool clinical instrument ophthalmologists use to check tear film instability. So the patient looks at this cone, and as you can see here, the concentric patterns are projected directly on the surface of the human eye, and the built-in software can track the stability of these individual rings to calculate something called tear film breakup time. Okay, so we used in placement of the patient, we actually used the device, and this movie is to show that we were able to form those concentric patterns. And we were able to do analysis to show very significant differences between normal and DED. So we saw much more rapid tear film breakup, and also this tear film breakup happening over a larger surface area. This is another clinical diagnostics. If you suspect that or the doctor suspects that you might have DED, then one way to do it is to actually put a drop of fluorescent solution, shines cobalt blue light on it. If your ocular surface is healthy, this is what it looks like. But if you have dry eye disease, then this is actually what it looks like. So desiccated tissues and cells end up taking the fluorescent molecules and fluorescing under this excitation. So we did exactly the same test to show qualitatively very similar to the human case. Ocular inflammation is one of the key pathophysiological processes in dry eye disease, and so we actually measure cytokine levels in the tears we collected from the devices to show that the DED model showed significantly elevated levels of cytokines over time. And towards the end of this study, we actually also partnered with a pharmaceutical company to test this drug called Lubricin. So as some of you may know, this is a natural lubricant found in synovial joint fluids, but this company is trying to kind of exploit this lubricating properties of lubricant to develop ophthalmic lubricants for dry eye patients. So we show that when we administer this drug, the drug molecules stick to the ocular surface, and importantly, as a result, it actually shows very significant friction lowering effects. And using those clinical measurements, I just talked about, we showed significant improvements in tear film stability and fluorescent corneal staining. But more importantly, we actually found out that lubricant treatment, reduced expression of TLR4 and NF-kappa beta, and also a reduction in the production of cytokines. So this was somewhat unexpected. So this platform allowed us to confirm the intended therapeutic efficacy of lubricant, but it also allowed us to discover this unexpected therapeutic effects in this study. Yeah, just to quickly walk you through some of the other disease models under development. So we have the model of the back of the eye. So a retina model, we're actually using patient stem cell derived cells to model age-related macular degeneration for drug testing. We also have a couple of lung models, lung disease models. This is one example where we engineer this tissue using asthmatic cells. We can compress the entire tissue in the lateral direction to mimic bronchoconstriction, airway constriction during asthma attack. So in this paper, we're actually showing that those pathophysiological mechanical forces, induce airway fibrosis and increased vascularity in asthmatic lungs. More recently, we also created this simple technique to engineer linear tissues, and using this system, we modeled cancer cachexia, muscle wasting. So we co-culture these constructs with drug-resistant tumor spheroids to show oxidative injury in muscle, and we use this system for drug testing. And most recently, my lab got $8.3 million grant from BARDA. It's a big grant to use lung-on-a-chip technology to study adverse health effects, especially respiratory effects of chlorine gas. So we're actually modeling acute lung injury due to chlorine exposure in this study. So there's a lot going on in the lab, but I guess from your perspective, a more important question to ask is, what else is going on in the field? So I was saying over the last 3 or 4 years, my lab has been at the forefront of developing new techniques, new ways to engineer more complex, more realistic tissues, to improve the predictive capacity of those organ chip systems. And so this happens to be also one of the main trends, recent trends in the field of organs on chips. And so what I'd like to do is to spend some time talking about some of those newer model systems we've developed recently as part of this effort. So the first example I'd like to talk about is to engineer blood vessels. So about 4 years ago, my lab got this award from Cancer Research Institute to develop cancer immunotherapy on a chip model using organ chip technology. So we wanted to see how engineered immune cells, injected into the body, interact with cancer cells in the tumor microenvironment. So in this work, very first thing we had to do was to engineer perfusible human blood vessels that can be controlled in microfluidic devices. So this is the device that we use. And this is a cutaway view. There are 3 compartments. The key design feature here is that there are 2 barriers protruding from the ceiling of the channel. So using surface tension, or capillary pinning effect, that's what it's called. It's a well-known phenomenon in physics. So you can inject liquid, and then you can trap liquid only in the middle compartment, and then you can gel it. So in this case, we're injecting hydrogel solution mixed with a fibroblast and endothelial cells. After gelation, you can fill these side chambers with media to feed the cells for 3D culture. So interestingly enough, actually, over time, in the presence of all the supporting factors produced by fibroblasts, these endothelial cells assemble themselves into 3D vessels that open to the side channels. So this is actually kind of, we're mimicking the developmental process of vascular development here, vascular genesis, sorry. So this is actually a movie of vascular genesis happening in this device. It's a SPATA movie taken by a phenomenal engineer in my lab, Andrei Georgescu. I hope you can see the red cells. These are RFP expressing endothelial cells embedded in fibrin gel over time. So over a period of 7 days, these guys connect with one another to form 3D vascular structure. So we can engineer really nice, micro capillary level vessels. And it's really interesting to see those fibroblast mesenchymal cells wrap around those vessels as if they're parasites. Like I said, these are small vessels, but they have open luminal compartments. And this is a scan electron micrograph of an actual microengineered vessel in our device. We can do TEM. So, we can, it's very similar to in vivo, structures down to the submicron levels. Oftentimes these capillary vessels at cell-cell junctions, they actually show this as cytoplasmic folds, but we see the same thing in our device. And I think that's because we're mimicking the developmental process in this, system. These vessels, qualitatively, you can tell, these vessels are different. These are good. These are not good. But it's really difficult to quantify. So we're actually using machine learning-based algorithm to segment, extract those features, and we're actually using this pipeline for more quantitative analysis of 3D vascular architecture. We can also tune the architecture depending on how many cells you put in, how stiff the gel is. And these are exactly the same type of endos, but they show the very different architecture. But the best part is they're perfusible. So we can perfuse these vessels with fluorescent dye, as you can see in this movie, or blood cells. So, red blood cells shown on the left and white blood cells shown on the right. Yeah. So we're now using this as a platform, to build more complex, physiologically relevant, realistic tissues. So just to quickly show you a couple of examples, we have a pancreatic islet on a chip. So these green clumps are donor islets, cadaveric islets. We get them, we put them into our device to vascularize, and we can maintain their function and viability for over a month. And we can also induce adipogenesis of stem cells concurrently with the vasculogenesis to mimic white adipose tissue. And this is actually cancer tumor explant, patient-derived, patient tumor explants that we vascularize in our platform. So just to kind of, help you better understand how these systems are constructed, I'm going to actually talk a little bit about this, one of the newest models in the lab. It's actually a model of human bone marrow, more specifically the hematopoietic vascular niche of the marrow. So the design inspirations from, for this model come from the process of bone marrow development. So as some of you know, bone marrow actually develops towards, the later stages of embryogenesis, and those hematopoietic progenitor cells, hematopoietic stem cell progenitors are generated at hemogenic sites. But during embryogenesis, they move around a lot, passing through these different embryonic niches that provide a lot of signals for expansion and maturation of these cells. But eventually, they actually migrate into this medullary cavity of the bone. They colonize the marrow. But studies have shown that this process occurs concurrently with the formation and maturation of sinusoidal vessels in the same compartment. So we wanted to model this concurrent process of HSC colonization and vasculogenesis in a microfluidic device. So we used this exactly the same device, but the content was different. So we actually filled this middle compartment with gel, but in this case, we embedded the CD34 positive human hematopoietic stem cells along with endothelial cells and mesenchymal cells. And what we did was we let them sit there for 2 weeks, over 2 or 3 weeks, they actually kind of self-organize into these very complex tissue constructs that are reminiscent of, hematopoietic vascular niche in the bone marrow. So this is a movie of this, construct after 3 weeks of culture. I know it's not easy to see, but there are a lot of vessels, and you also see these very small, teeny tiny cells moving around a lot. So there's a lot of cellular activities and also hematopoietic activities happening in this device. We show that, definitely the gel is colonized by HSCs over time, but, we can, do directed differentiation and by changing media conditions to generate white red blood cells or white blood cells. And some of these white cells are very, very mature, as you can tell from these morphological features in neutrophils, for example. We also show that we can maintain the stem cell population in this long-term culture system. We've also developed a technique to extract and harvest these devices after we form them. So with the single-cell RNA-seq to really dive into the biological complexity. So in the interest of time, I'm just going to skip over this, but we found out that we start with 3 different cell types, but we end up with more than 10 different cell types, expressing different markers. We also looked at the trajectory, the single cell trajectory analysis to look at the stem cell differentiation into these different types of cells and how these markers actually change over time. We also conducted very complex, ligand receptor interactions to look at some of the known and also unknown, signaling pathways that are present in this model system. So what was more interesting is actually, so when these white blood cells, for example, are made in the system, as you can see here, these white blood cells are shown in red here, they actually remain in the perivascular region, but we can actually stimulate them and get them to intravasate, as you can see here, using cytokines and drugs. I have to skip this, but we actually also modeled the hematoablation, bone marrow ablation using ionizing and radiation. So probably, this is actually a more exciting thing to talk about. So in this paper, in the last figures, we're actually presenting this multi-organ system consisting of bone marrow connected to the lung on a chip. So this lung on a chip, kind of similar to what I talked about at the beginning, fully differentiated epithelial tissue, stromal compartment, and vascular compartment. So the first thing we did was to infect the lung with bacteria. So a lot of interesting things happened, but most importantly, they actually went crazy and started producing a lot of chemokines and cytokines that flowed into the marrow, and then they actually induced massive release of neutrophils from the marrow. Then these guys intravasate and flowed into the lung to do what's shown in these scanning electron micrographs. So first they stuck to the endothelium. They actually were fortunate enough to capture these moments of endothelial transmigration of these neutrophils. This is right before, this is right after. They come out on the lung surface, they migrate towards the infection sites, and they actually start eating up these bacteria. We even saw netosis. These guys are like Spider-Mans, like they eject their intracellular structures to capture and trap those bacterial cells. We also saw neutrophil swarming that happens when they fight up large number of pathogens. So I guess what we're presenting here is a self-healing device, if you will. So there are no human interventions except infection. In response to infection, this model makes it possible to mimic the entire process of immune cell recruitment in this system. Yeah, so I think that's what's interesting about this platform. So actually we built the system to for this project. And so we send this chip to space to study immunosuppression in microgravity. So it's been known for decades that astronauts get sick more easily, somehow during or right after space flight. And so there's, kind of anecdotal evidence, suggesting compromised immune function in microgravity. So we wanted to send those chips to space and then infect the tissue on board the ISS to see what happens. I have to say it was epic failure, actually. So on the way up, this box actually, so it made it up. And we even took a photo, the astronaut took a photo and sent it back to us. It was exciting, but the flow controller was short-circuited on the way up. So it was, when we actually heard the news, my face kind of looked like this, So, but it was a good outcome was, we actually, got to really develop this really exciting, model system. Yeah. So cancer immunotherapy is another, kind of area we're venturing into, and using this platform, we can use tumor spheroids or explants to make these heavily vascularized tumor constructs. This is another platform. Sorry for flying through these, very quickly, and I would like to just meet the, the timeline here. So in one platform, we actually created this open-top platform where we first put the plug in, and using the technique that I just explained, we would form the vessels. And when we get a call from our collaborators, okay, the explants are ready, then we go pick them up, we open it up, drop them in there to vascularize. So you can actually, the beauty of this is you get to really control where these, tumor constructs, explants end up. And because these vessels are perfusible, we can directly perfuse the tumor constructs, using cells, media, drugs, whatever you can think of. Yeah. So, using this platform in collaboration with two physician scientists, Steve Abelda and Edmund Moon at Penn, we're actually modeling and studying CAR T cell trafficking in solid tumors. So we have a lot of data, but just kind of show you a quick snapshot of this project. So this is actually a lung tumor explant engineered to, this is a xenograft model tumor explant. So these lung explants overexpress mesothelin. This is a tumor antigen. And these CAR T cells are engineered to target this antigen expression. So this is a couple of hours after infusion. As you can see, many of these T cells stick to the tumor-associated vessels. And then over a couple of days, they infiltrate into the tumors that are found within the tumors. And as a result, we saw a very significant effect. And we actually did a control where the tumor cells did not express or expressed very low levels of mesothelin. You can appreciate the fact that there's a lot more trafficking in the mesothelin expressing tumors. And we also saw very significant effects on tumor growth as a result of CAR T therapy. Let's skip this. And a nice thing about this, model is that we can actually, because of the open-top access, we can gain, we have direct access to the tumor constructs. We can harvest, isolate those tumors, and then we can use those samples for further analysis. For example, we did very thorough flow analysis, flow cytometric analysis to look at differences, between T cells in the tumors and those in the surrounding tissues. And more importantly, we did single-cell RNA-seq to really dive into, the complexity of the interactions. And so, unfortunately, I don't have time to go over these, but we saw, we used the single-cell RNA-seq to really monitor phenotypic changes that occur in those T cells as they infiltrate into the tumor microenvironment. Yeah, I'll just skip all this. Okay, and this is another really exciting research thrust in my lab. Over the past year or two, we've been exploring the possibility of incorporating organoids into organ chips as a way to, develop more realistic, more predictive, physiological relevant model systems. So the idea is to use microengineered devices to tightly control the 3D culture environment of organoids for the purposes of, for example, increasing the lifespan of organoids, or for the purposes of pushing them more towards, mature tissues. So this device is shown here called Octopus is an example of such devices. This is a paper under revision in Nature Methods now. And so to help you better understand what we're trying to do, this is actually how people, traditionally, one of the most traditional methods to form organoids. So you can form this drop containing stem cells, and over time, in the presence of this three-dimensional environment cues, those stem cells self-organize into those, mini organs. In this case, I'm showing the self-organization of intestinal organoids. But there's a problem. So if you don't pass these cells, up to 5 days, they're good, they're viable, active. But over time, they die off, because one of the reasons, most important reasons, is actually you end up, with this necrotic core at the center of the, the drop. And over time, all the cells and organoids die off. So with that, wouldn't it be nice to be able to kind of, remove this core? This is a conceptual representation of what we're trying to do. Get rid of the hypoxic core, and then radially segment this shell structure and lay flat, to eliminate diffusion limitations. So this is actually the device that we came up with, Octopus. It's an acronym for this long name here at the bottom. It's actually designed as an insert, and it has a central port and open chambers. And so you inject material, and then it's distributed into these 8 chambers that are open. So and then constructs are much thinner. So there's, limited actually, or the minimal diffusion limitations in these systems. They have a movie here, and just showing the convenience and ease of, using this system. So these are designed as inserts, as I said, and all you have to do is the same thing you would do in drop culture. You would inject hydrogel into these, loading ports, and you would form these constructs, for long-term culture. So using this system in collaboration with Kate Hamilton at CHOP, we got some intestinal stem cells, from ileum, terminal ileum of healthy subjects, to show that in drop culture, they die off. In our system, they thrive. And it's more than that. We can actually grow them without passaging continuously over 1 month to make these massive, organoids. Of course, we've shown that they're more mature using these different markers of, differentiated cells. We also use single-cell RNA-seq to really show that, this method gives you better, more mature organoids. I'm sorry for skipping all the details here, but we also use this platform to build a disease model. In this case, we create a model of inflammatory bowel disease using those stem cells isolated from those patients, to show that, there are things that happen actually only in the system that don't happen in drop culture. We also use single-cell RNA-seq to discover these long non-coding RNAs actually that had not been described in the context of IBD. And we modeled intestinal fibrosis. Spontaneously, they become fibrotic, because of these pro-inflammatory cues produced by the diseased tissues. And in this last figure, we're actually, showing the vascularization of organoids. And we can do really interesting things, like, for example, we can flow monocytes, and then we can mimic their extravasation, accumulation in these inflamed tissues. So we can, we're hoping to model other interesting things that happen at the parenchymal vascular interface using this platform. Just a quick mention, we've been doing a lot of work in the area of reproductive biology and medicine. So we published some papers about placenta on a chip. Cervix on a chip is in the works, and this is a new paper coming out in Nature Communications, where we modeled implantation of embryos during pregnancy. So before I finish, if I may, over the next couple of minutes, just a couple of minutes, I'd like to talk about something that I think is really exciting that's happening in my group. So hopefully I was able to convince you that this is a compelling technology using those examples, but there is actually inconvenient truth that I like to say we, but I'm not sure if all the organ chip engineers would agree. I think there is inconvenient truth that we as organ chip engineers don't like to talk about. So it's really great that we can mimic complex things that happen in the body using these systems, but complexity is also what makes it really challenging to translate and scale up this technology. So simply put, it's not easy to make these devices. It's not easy to use them. Oftentimes we have to culture cells, maintain these tissues for long periods of time. Something goes wrong and we have to start all over. And so for these reasons, it's been really difficult to scale up, this technology. And like I said, so in other words, it becomes really challenging to conduct many, many experiments concurrently to, get data. So it really limits our ability to get, data from these, what is supposed to be, like really good model systems, right? And oftentimes creating these devices requires at least half a dozen, steps, that are really prone to human error. And so there's a lot of variability, lab to lab, person to person variability. These techniques are not easily translatable to other labs, other users. So we wanted to, we've been trying to solve these fundamental challenges and problems. So first we've been trying to scale up by walking away from these devices. This device, one unit, but we've been using those plate devices, each containing tens of replicates. And more recently, this is the most advanced, platform we've built in the lab. So this device contains 128 individually addressable, fluidic chambers, cell culture chambers, designed by, again, another, it's a phenomenal student in my lab, Andrei Georgescu. We actually use a combinatorial, fluidic circuit actually to control, fluid flow and fluidic environment in these systems. This is microfluidic valves in action in this system. And this movie really shows the individual addressability of the system. So those rectangles shown on the left are cell culture chambers. And you can really see, using this system, we can really, determine and control what goes into each of these chambers in a precisely controlled manner. We've spent a lot of time and effort actually trying to automate the manufacturing process. And so initial version of this platform had more than 2,000 holes to punch. And, it took Andre, I guess 4.5 hours to do that manually. So he got really, frustrated and he built this punching robot from scratch. So this is actually a robot in action. It cuts PDMS like butter, it uses rotating spindle. And between punches, the ejection pin, comes out to push out the core. And this is real time. So, we can punch over 2,000 holes within, 20 minutes very quickly. But it also allows us to really, do this consistently, and it really improves the quality of the hole punching, as you can tell by comparison here. We have very fine precision, spatial precision, and we are using and building robots to automate fluid handling. I think this is very important. So this is actually one example where we are actually using the robot to load the mixture of cells and hydrogel into the blood vessel forming device that I talked about earlier. And then we can also automate many of those steps, of cell culture. So going back to where I started, it's really, these robotic systems, automated systems are making our lives easier, but it's more than that. We're most excited about the data. So we have the ability to now dramatically increase the amount of data we can extract. So one device gives you 128 experiments. We can easily stack 5 of those. Then the concurrent experiments, the number of concurrent experiments easily goes over 1,000. So this is actually, in one example, we ask questions like, under what conditions can we engineer most physiological, realistic pancreatic microvessels? And so we are able to answer this question using this platform, high throughput culture capabilities. So I'll just, quickly move on. But so based on this vision, we started a company called Vivadyne, with this seed funding from VC. And so we're trying to develop a whole pipeline consisting of, the system, also automated, instrument, and also including software as well. So we're pretty excited about this, vision that we have. Okay, so that's what we do. There's some papers and talks, you could watch about the potential of this technology for pharmaceutical applications. And lastly, I'd like to thank my group and my collaborators and funding sources for their help and support on some of the studies I talked about today. Thank you very much for your attention.
And thank you for the fantastic. Sure, I think everyone can hear me better.
Yeah, but that's people online.
All right.
Oh,
Some
it
people
was
are like, yeah.
awful the whole time.
Okay, so thank you for the fantastic talk.
Thank you.
So we still have time for some questions. Let me just say quickly that this series continues. And next week on November 5th, we have Milan Jaeger, who is the Executive Director of the American Institute for Medical and Biological Engineering. And I would just like to invite everyone here in audience and online to follow us on social media. This is where you can find all information and you can register for different BME seminars. So let's start with questions. We do have.
Yeah, we'll be doing a combination. I'll take care of the questions because I have the list here. We'll do a combination of. Yeah, I got this. Yeah. Okay. We'll do a combination of questions from the audience as well as people in person.
Sure. Yep. I could try to repeat the question. Yeah. But, yeah.
All right, Dan, that was great talk. On your lab on a chip where you had the bone marrow and the lung, with the lung infected, does the bone marrow response actually clear the infection if you leave it long enough?
So that's a good question, actually. So the longest we've tried actually in these experiments was about half a day. Yeah. So we did see a significant reduction in the number of bone marrow, but I don't think we saw clearance of infection in this system. But it would be an interesting experiment to try. Yeah. I believe that we have the ability to, and because so I guess the production rate could be one question, like how fast, those neutrophils be produced by the marrow on a chip. But we do have the ability to maintain these tissues for long periods of time, over a month. And so maybe not on the same time scale, but maybe over longer periods of time, it might happen. Yeah, but interesting idea. Thank you.
We had a question from Diksha Hegde online. How extensively do you think organ-on-a-chip models will be used in the future? Where do you see the field going in 10 years?
Good question. So I guess the answer would vary, depending on who you ask. But, the stuff that I talked about towards the end, it's just, I don't like the fact that, so organ chip, this is my personal opinion, has been a huge success as an academic field, academic research topic. But I think we're actually trying to make a real impact on, practical applications like drug testing, happening in pharmaceutical companies and academic labs. But I just don't like the fact that these systems are developed and they never get out, So that's actually what we're really trying to do as a way to really enhance and, prove the true potential of this technology. So I think, and also, Gordana in your department actually is making a huge impact on this collective effort as well. So there are, labs trying to make this happen. And I hope, and I think we will see some meaningful outcomes, from this collective effort over the next 5-10 years or so. Yeah. And as I said, during my presentation, increasing the complexity is also one of the things that many people are trying to do. But if you think about it, you want to be, approach this very carefully, because complexity of the model is, I think, dictated by the question you're asking. So it's really important to start with, good questions in, biology and, clinical medicine. And because, we're only mimicking certain aspects, that's actually one of the things I should, improve upon. Like I tend to kind of overemphasize and really oversell these technologies and model systems, but we're only, mimicking limited aspects of, very complex things that happen in the body. But, our ability is evolving, improving, So I think we'll see more, complex, realistic model systems, appearing, emerging in the future. And then really, using these systems for clinical trials and, for practical applications and, drug discovery, I think that's going to be really important kind of, a goal for the field. And also really interesting kind of, cross-fertilization between organ chip, field and then development of biology. I think especially with this idea of organoid on a chip, I think that's also an exciting kind of avenue to explore in the next 10 years.
Hi, Dr. Hunt. Thank you so much for the talk. I really enjoyed it. So I had a question. You mentioned the inconvenience of this whole system and how it's so hard. If you mess up one step, the rest of it is kind of like you have to start from scratch again. So you also talked about automation. And so I was wondering, like, why isn't automation being implemented more? And will this change the landscape?
Yeah, so I believe, automation might be, I don't know. It's just, I guess we're trying to achieve high throughput and automation at the same time. That's what's, what makes this, kind of whole effort very challenging. So if you're dealing with, kind of small replicates, small numbers of replicates, automation may not be necessary, but still, I think that, all these, different steps are so prone to human error, And so I think it's, I mean, we're actually working on this paper where we're showing, the variability, of model systems, in manually produced systems, automated systems. We definitely see huge improvements, in those automatically generated systems. So if you have access to those, kind of expert facilities or, instruments and also expertise, of course, we're not robotics lab, right? So we work with roboticists and, engineers to make, those changes happen. But I strongly believe that automation might be one of the ways to go. Yeah. So let me actually put it that way. But
Thank you so much.
Hi, just to follow up from your previous question regarding automation, do you believe that this is kind of the way forward for academia, where we should train all of our PhDs and postdocs to do the same? And is that something that you're trying to strive for in your lab?
Yeah. So one thing you want to think about is actually, so Dorales, who are you asking this question? It's like, or the postdocs and student trainees you're talking about. Are they engineers or biologists? I guess that really changed the answer to this question. So if you're asking a question for postdocs and trainees in biology, medical, kind of pure biological science or biomedicine, yes, I think it'd be great if they could have access to those automated tools and more kind of tractable systems. And their focus is going to be on the data and then insights actually gained from these experiments, right? So I think I believe we would be able to play an enabling role in those, types of studies. But if those trainees are engineers, then that's a different story. Because, we as engineers, I'm an engineer, so I'm genuinely interested in developing new platforms, new model systems, new designs. So by using those systems, by actually being asked or forced to use those systems, that would suffocate the creativity and, academic activities. And so in those cases, I think, there are going to be always, groups of engineers and, trainees, focusing on developing new designs, new systems. But important thing to think about is if you had a nice platform that could accommodate those different designs being developed by, academic labs. So that's actually one of our visions, goals for the company. So you can abstract away from these, designs. You have a platform that can accommodate those systems. So, if you have something like that, then, I think that would strongly also support, those engineering efforts, focused on developing new designs. I hope that answers your question.
Thank you.
Yeah.
Thank you. Okay.